Now we will see the second method, that is Edman degradation. So in 1950, Pierre Edman, they developed a method for the protein sequencing. So this method was developed in 1950 by Edman. So that's why the name for the sequencing is given as Edman degradation. So it involves sequential identification of amino acids from the N-termini. So as in the Sanger sequencing, we are determining the amino acid sequence of that particular protein from the N-terminal. So likewise in here also we are determining the amino acid sequence from the N-terminal end of the polypeptide chain. Then for this, phenyl isothiocyanate PTC reagent is used for the Edman degradation. So sometimes it is also called as Edman's reagent. So phenyl isothiocyanate. Then the amino terminal, that is N-terminal residue of the protein, can be identified by reaction the protein with the PTC that forms a stable covalent link with the free amino group prior to hydrolysis with the 6 molar HCl. So again, we are using the phenyl isothiocyanate over here. We are taking our sample and we are adding this phenyl isothiocyanate. Then what will happen? It will react with the N-terminal end of that particular protein and it will form a stable covalently link with the alpha amino group complex. And after hydrolysis, again, as that of the Sanger's method, we are carrying out the hydrolysis and we are determining the sequence of that N-terminal amino acid. But before processing the Edman's method, so two things are very important. First, we have to break the all non-covalent interactions in case of oligomeric proteins. So if oligomeric protein is there, then there may be different subunits will be present. So we have to separate these subunits by breaking the non-covalent interaction. Then how we will remove these non-covalent interactions? We will use high concentration of the urea over here. Then we can use guanine hydrochloride over here. So by using these chemicals, we can break the non-covalent interactions. Then second thing, as we are carrying out the Edman's reaction over here, we are taking our sample and we are adding this PTC reagent. So what will happen? It will carry out the sequencing process until it is achieved with the disulfide bond. So that means if the disulfide bond is present into the protein sample, then that time afterwards it will not work. So that's why we have to break the disulfide bonds. Then how we can break these disulfide bonds? So for this we can use the beta-mercaptoethanol. So beta-mercaptoethanol will reduce the disulfide bonds. And as we know that the have to block so that again they will not reoxidize and again disulfide bond. So we have to block these free cysteines that is present into the protein sample. Then how we can block this? Or we can carry out the reduction by dithiothreatol, that means DTT, and it is followed by acetylation by using iodoacetate. So if we use these chemicals, then that time these free residues will get blocked, and after preparing this we can carry out the Edman's reaction or Edman's method. Then this phenyl isothiocyanate react with the uncharged N-terminal amino acid group of the peptide to form a phenyl thiocarbonyl derivative. So PTC derivative peptide will get formed due to the reaction with this PTC with the N-terminal amino group. Then under mild acidic conditions, this cyclic PTH phenyl thiohydration of the terminal amino acid is liberated, which leaves an intact peptide that is shortened by one amino acid. So that what will happen, we are again carrying out the acid hydrolysis, then that time this PTH or of the terminal amino acid, that means that particular derivative will get liberated, and which will leave the peptide which is shortened by one amino acid. So that means we get the PTH amino acid derivative of the N terminal amino acid. So it will get liberated, and we have to take it into the, or we have to extract it, and afterwards that particular polypeptide chain will be there, which will contain one less amino acid. That means terminal amino acid will get released. Then this released PTH amino acid is identified by high performance liquid chromatography. So this we have to identify it by using the HPLC. Again, here we are using the different standards of amino acid, and we are taking that particular released PTH amino acid, we are carrying out its HPLC. We are comparing the peaks of this amino acid with the standard amino acid peaks, and from this we can find out which amino acid was present at the N terminal end. Then next, the sequencing techniques has been automated and refined so that upwards of 50 residues from the N terminal of a protein can be sequenced from picomole quantities of the material. So at a time we can sequence up to 50 residues of the amino acid from the N terminal end. So that means if we have to sequence the protein by using this Edmonds reagent method, then that time our requirement should be, it should be a 50 residue chain. So beyond this, we cannot determine the sequence of the protein. So here you can see this is the chain of amino acid. That means this 1, 2, 3, 4, 5 amino acids are present into that particular polypeptide chain. Then next we are adding this PTC, that means phenyl isothiocyanate, into our sample. Then our sample will be labeled with the this PTC reagent. So afterwards we have to carry out the hydrolysis. So under acidic condition, anhydrous trifluoroacetic we have to add. Then afterwards thiazolone derivative will be formed, and a polypeptide chain which will contain one less amino acid will get formed. So we can get two products. First is thiazolinone derivative and peptide chain which will contain one less amino acid. Then we have to extract this thiazolone derivative in the organic solution. Then we have to treat it with the aqueous acid, and we get the PTH amino acid derivative. So here you can see this product, this PTH derivative of the first amino acid which was located at the N terminal end will get formed, and this we have to identify by using the HPLC. So we have to compare the peaks of this PTH amino acid derivative with the standard amino acid peaks, and from this we can get to know which amino acid was present at that particular C terminal end of the protein. And here we can get the peptide which is shortened by one amino acid residue. That means one amino acid was used over here, and the peptide chain was released, so which was having one less amino acid. So as I have told you that the limitation of this technique is that up to 50 amino acids sequence we can determine by using Edmonds reagent. So for this, how we can get the short sequence of the proteins? So for this we can treat the protein via two ways. First, we can carry out the enzymatic degradation. or we can use the different enzymes, or secondly, we can use certain chemicals. So enzymes such as we can use the trypsin agent. So it will cleave the carboxyl terminal of the lysine. That means if the lysine residue is present or arginine residue is present into the chain, then after that residue, that particular polypeptide chain will get cleaved. So likewise, different enzymes are present. The next example of the enzyme is chymotrypsin. Again, this chymotrypsin will cleave the amino acid residue that are aromatic. That means phenylalanine is there. So such after getting such aromatic amino acids, the chain will get cleaved. Then apart from this, we can also use certain chemicals. So cyanogen bromide is there. So that will cleave the polypeptide chain after the methionine residue is there. So likewise, we can use enzyme, or we can use the chemicals. So by using this, we can reduce the length of the polypeptide chain and we can sequence the protein. Then so this type of the sequence we can get from the sequencing technique. So here you can see the glycine, phenylalanine, then lysine is there. Then second is threonine, phenylalanine, lysine is there. So we can get the different fragments and we are sequencing these fragments. So next step is we have to compile this data. That means we have to find out which overlapping sequences are there, and we have to compile this data, and we have to find out the what was the sequence of that particular protein. Then next are the limitations of Edmonds reaction. So Edmonds reaction process from N-terminal of the protein. So it will not work if the N-terminal amino acid has been chemically modified. So if we have modified the N-terminal end of that particular polypeptide chain, then that time Edmonds reaction will not work. So this is the limitation of Edmonds reaction. The next limitation is that it is required to use of either guesswork or separate procedure to determine the position of disulfide bridges. So as before starting the Edmonds reaction, I have told you that two things very important over here. First, we have to break the non-bonds, and then we have to break or remove the disulfide bonds. So if that particular protein will have the disulfide bond, then afterwards beyond this reaction will not work. So we cannot find out the sequence of the amino acid. So this is the limitation of this technique. Then next are the applications of protein sequencing. So by using the protein sequencing, we can get the knowledge of the sequence of amino acid in a protein, so which will offer insight into the three-dimensional structure of the protein. So by using the structure of the protein, we can get to know the function of the protein, and we can find out the cellular location of that protein. Then certain amino acid sequences serve as signals that determine the cellular location, chemical modification on a half-life of a protein. So there are certain amino acids available, so which can signal the cellular location, chemical modification. So that we can use or we can find out. Then next, protein sequences can elucidate the history of life on the earth. So by using these protein sequences, we can find out the history of life on the earth. Then apart from this, we can identify the protein family to which a particular protein belongs. Then we can find out the sequence of the gene. And we can also discover the structure and function of the protein by using various computational methods and experimental approaches. So this is regarding the protein sequencing.